key part of this is that it's a data management system. So we're focusing on a lot of the data associated with photo ID. So as you all sort of realize, photo ID is hugely time consuming and visually fatiguing, and we wish we just really didn't have to do it at all. But it's subject to human error, and when we're going between different photo ID projects, it also, taking that cross comparison takes a huge amount of time as well. So to help alleviate some of the time consuming part, there's been a number of systems developed in the last 15 years. And this is an example of the FinScan that was developed in the late 90s, where we tried to use, look at dorsal fins and identify what the notches are and come up with different algorithms to look at that. But these have only focused on the matching part of photo ID and are usually very species specific. And they're not completely automated, so that's why they're computer assisted. They basically require the user. And in some ways, like FinScan or Darwin, they could also increase potential time to process photo ID because you need to play with a lot of the image segmentation because they're not perfect. So there's a lot of time required as a part of that. But there's so much other data associated with photo ID. Not only the life history parameters associated with the individuals, but you also have a lot of sighting information that goes into group size, spatial patterns, etc. And a lot of this has not been the focus of different software. And because those other softwares are so species specific, there's so many other different species that there's no software available for. So we tried to develop an overall comprehensive approach to store, visualize, manage, and analyze your photo ID data. And we realized that there are really no standards in the field, and what John Moran presented yesterday and how he processes photo ID is totally different than another research lab that may process photo ID. They do image quality in one way, we may do it another way. So there's different methods in photo ID how to process. So we really tried to make this a dynamic system to change over time, and also whatever your research project's needs are. And we realized that we're getting more and more long-term data sets, and so this really requires collaboration between different research groups. And so we really need to try to standardize databases, because doing those comparisons between different databases from different labs is hugely time-consuming as well. So as we were creating this, we also realized, we don't want to reinvent the wheel. So we tried to be inclusive as much as possible and not exclusive of other tools. So we tried that as much as possible. And we realized, for one species, there can be multiple catalogs. For example, with gray whales, just on the population of western gray whales, we have 4 different catalogs that we all have to interact with. And we all have different identification numbers. So we can interact with them, and also for the eastern gray whales, there's a number of different catalogs. So if we really want to understand the species and the individuals, where they occurred throughout geographic range and throughout their life history, we really need to take use of all those different information. So we need to extend collaboration. But even with one single study area, for example, in Kaikoura, I can go out in one day and identify 5 different species, from sperm whales, dusky dolphins, common dolphins, killer whales, Hector's dolphins. And this presents a huge management issue of how you deal with the data with multiple species in one single area. So that was our sort of approach with discovery. And I will transfer to my colleague, Leszek Marski.
Yeah, this was the approach in discovery, and this chart represents the conceptual framework of discovery, where from field data collection you go through all this pre-matching and matching procedures to all the associated information, sightings info, and survey information, with any potential additional information, acoustics, telemetry, genetics, whatever else is being collected during the field work, and then all assembles in this data management component the core part of discovery, and then a number of those outputs and summary of the results. So we'll go step by step very briefly. We cannot cover the entire range of components, but the main parts to explain the flow and concepts of discovery. So as Glenn mentioned, different researchers do things differently, and we need to, there is a need for this dynamic system that we can incorporate all these individual preferences and choices. So in this case, the discovery starts from the very beginning with very user-specified dynamic systems, where we actually specify all the things you want to have in your system, starting from study area, from you can include your maps, you can choose, or you include the species, you can have multiple species within one database. You go to your individual classification, and you can choose what you will be using for your classification, features you will be using, what type of descriptors. It's a non-index based system. We'll be referring to this number of times, so it's a number of those classifications aspects are categorical. All the associated citing informations, environmental, group structure, group composition, age, classes, and so on, behavior, and all the miscellaneous informations that actually you define what you want to include there, or anything that will actually be observed during the survey that you cannot predict ahead of time, and it comes, you don't want to get it lost. And since we all have habits, and habits are sometimes good, sometimes not, but they're very, very hard to get rid of. So some people would have certain preferences of putting data in a certain way. Even in simple things like whether to use decimal point or minutes and seconds in geographic position, whether to use original images, whether to use temporary IDs, or perhaps to verify. If you're just a single researcher running with one project with one or couple of catalogs, you might feel that you don't need to verify temporary IDs, although it's probably good to verify them no matter what. But if you work with multiple teams, the verification is critical to avoid mismatches. So all this can be incorporated and is user defined. So going on with this fly chart, all this pre-matching and matching procedures. So first you start with file naming. As it was mentioned yesterday, and with all we know, file naming is pretty critical. You can have all this important information there. This process is automated in Discovery. You can actually choose the whole range of images and have them renamed as a batch process. And you can define how this renaming is going to be structured. In our case, we include temporary ID, date, study area, group size, group number, or encounter, or citing number, depends on how you use it, photographer, and the frame name. But again, this is user defined. You can decide how your naming structure is going to be. And then it comes all this tedious process of filtering and processing the images, potentially cropping, looking at those parts which are really important, that have all these identifiable characteristics. So if we have an image, like in our case with the work in Africa with elephants, well, you select this part that you really want to, that contains the information. You can get rid of all this background. And actually, you can still improve the image to some degree. We have some tools here that you can increase the contrast, brightness. You can sharpen the edges, and this really helps. Particularly with out at sea, it helps a lot when sometimes just a little bit of extra focus makes those couple of notches more on a dorsal fin a bit more visible. So that's a very nice feature. And then you can plug this into different categories if you like. Quality, for instance, entirely user-defined. There's a number of quality criteria, quality scales. We in Hong Kong use a scale of 1 to 100. Some others use 1 to 6. Some others use basically 3 or 4 or 5 categories, how they're subdivided. That's your choice. You can create sections like, for instance, mother and calf groups, where you would want to keep a record of all the mom-calf associations and keep a record maternal care on weaning age and potential death records and so on. or we keep all the images. We of course use some to the catalogs, but we don't get rid of any. If they're unmarked individuals, we plug them into the unmarked section. And once you select all the individuals on the images, marked, unmarked, sometimes there will be several individuals on one image, then automatically there is a count going on, how many marked and unmarked individuals are there, so it calculates you the ID ratio, which is very critical in all the population size estimates, which comes very, very handy for those who potentially would forget that this is actually an important thing to keep eye on. It's not that unusual, surprising. And then matching within a group. Obviously, first within a group, within an encounter, as we call it, where you want to select your best representative images of each group. So you have your query image, and then you compare it to your selected images, basically to your catalog. And you can choose. You can use just one query image, or if you're really good, you can increase your number of query images. So you can make the matching to few individuals at the same time, if you know your animals really well. Matching can be done in horizontal and vertical mode. Depends on what species you work with. For dolphins, dorsal fins, you would use the horizontal mode. Whales are generally, as this case, gray whales are mostly used in a vertical mode. And again, query images and your sample from the catalog to verify the query image. And you can actually you can individually decide how many of the catalog images you want to have shown at the same time. So it could be 6, like for the dolphins. You can choose only use 1 or 2 or 8 or whatever you prefer. So you make this selections at the beginning in your settings of discovery. And in some cases, you can actually, if you like, you can use this thingy here. English is not my home language. And you can do individual zooming to pull out some of the specific features of so individual per image, not all of them, or not removing all the others. So, but it is non-indexed method, so that actually your eye is the final apparatus to decide what you deal with, and basically you search through the catalog by filtering and sorting the database. And once you find a new individual, like a newt in this case, from Hong Kong, new individual or match. Well, then you include all the individual information, such as your ID, your species, your group, your date of sighting, and so on, your time, if that's important. Your locations. Your location can be imported from the file or entered manually. Individual features, those that never change through the lifetime of the individuals. Obviously, sex generally doesn't. Day of birth doesn't. The age might change, but the day of birth will remain the same if you can estimate it. So that we will be included there. And your user-defined non-index classification, as well as all the miscellaneous user-defined informations, such as, for instance, your age class, which will be changing throughout the life history of the individual, your potential mark change, growth. If you work with small enough critters, they can measure how much they increase in their size and so on. So this will be all the pre-matching and matching procedures, and then we go to sighting informations. And our concept was that basically we took our data sheet, and that's just an example of our data sheet from Hong Kong, and we decided that all the information we're collecting, it's obviously important. So we want all this information that we have in a field data sheet to go to the discovery and be freely accessible at any given time. So any information that's there, which will include all this kind of bookkeeping for the survey, all the effort, time, photographic info, and so on, all goes in. All the environmental information, at least at the beginning and the end of the encounter, is in our procedure. All this stuff that comes, like the surveys of, let's see, sea state, depth, location, distance from the shore, habitat, and so on. Then I presume everybody that works with almost any mammals, certainly with marine mammals, you keep record of the group structure, group composition across the survey, which might change. So we would do this regularly through the survey. All this information will go in, along with geographic positions and environmental information collected through the survey, and the behavioral stuff. We would have another data sheet purely for the behavioral data, but in a summary data sheet, it will be at least some records of behavior throughout the survey. And then you just go and enter all this into the sighting information. So your group information, your age classes, number of individuals of individual age classes, your photographic data, what camera you used, who did the photographs, who took the notes, and your locations, again, either imported or manually entered, environmental data, any comments that you might have to this specific sighting. You never know what can come up during field work. And this miscellaneous, which in this case, in the example of the miscellaneous, will be the biopsy. Okay, so we took the biopsy, categorical, yes or no. If we did, whom did we sample? Was this the particular individual there? In your other file, when you enter your new individual, you will replace your in this slide just a little bit earlier. You would replace your temporary ID with a new permanent ID. But you can also assign potentially name if you want to use. If you're better with names for individuals, as I am. I never remember if it is 024, 035, but if it is John or Mary, I will know exactly who it is. So you can then have this information in your miscellaneous info, like the biopsy data, and it sits there forever. Along with all this geographic positions, all this information that we collect throughout the surveys, all the track data that can be incorporated, and goes along with every encounter data. And after this, the main component, the most complex, which again is going to talk about it, data management, which is really the essence of discovery.
So once you have all your data in, into a central database, you have a lot of procedures you have to go through, and you constantly are checking your database, re-verifying your IDs, and also looking at the individual information. And so you can see this is the catalog of your individuals, which gives you summary data of how many images, how many sightings, when did you first see that individual, when is the last time you've seen that individual. And then when you click on the individual catalog, you can see all the images associated with that individual. So these are the different re-sightings, and you can even have multiple images within a single day. So if you want to include, 100 individuals of the same individual within a group, well, sure, go ahead. But usually we only have to include one photo per sighting. But then you can also have an important aspect, I think, is a cross-link between different catalogs. So, for example, this is our catalog for gray whales, but what we call individual 083, another study calls individual 062. So it's a very important thing because they process their IDs, and if we want to make use of all the data, we have the important cross-link between the two studies. Then you have the summary information of your sighting records. And here you can partition your data by your study site if you want, or your species, or the date of study. And this can be, partitioned in any combination of ways. And for example, this is just one date where we pull up the information, and you can see all the sighting data that were recorded. So here's sort of the standard sighting information. And then for any of these sightings, you may have multiple records for each one. So in the environmental, as we're on this group, we had multiple records here. And then, as well as the geographic position. So we just click on a button there, and it pulls up that information. And so we can re-verify that those data have been entered correctly. And then we constantly want to review our database, look at it, and there's this, it's all in Access, so you can use Access if you'd like, or we have this interface here that also lets you visualize all the different tables you have in the database, and it has some QA, QC functions that help you identify potential issues. Like this is a function that goes through and says, oh, you know, you have some problems. You don't have those images on your actual physical disk, so you got to go find those images. So that helps to go through your data and make sure it's all there. So there's all the tables, and then check for directory. And then if you already have existing data into different processes, as you can do different imports. So here's importing your images into a database, which can use the file name structure. This is sort of a quick import. Then we have a more complex, versatile import, where if you have information in your EXIF part of your data, you can put that like date and time or geographic position. You can automatically extract it from your EXIF data. Or if you have the information like ID or date as part of your file name, you can put that information into the data, and it automatically enters into the system very rapidly. But then a lot of people already have data in different forms. We have our import for discovery databases, which if you have multiple studies that are working with discovery, you can just import them in really quickly. Or if you have your data in a different format in Excel or Access, you can also use this dynamic import, which says this is the different fields, and you can include just one value if the study area is always the same. You can just put Hong Kong or whatever, and then you pull in different columns from your different databases, and then import that into Discovery. So another important part is once you have your data, and the real power of having all your data into one centralized database is that you can do really cool things and start looking at your data visually. And this is our GIS mapping system that we incorporated to just really give you a quick look of what your data are and what you had. And this is an example of one survey for Indo-Pacific humpback dolphins in Hong Kong. And you can see we have multiple layers, and all of these are shape files with attributes. And you can see with this survey, you have the individual records where we took photographs of these different individuals. And then you have the sighting locations, when we started, and every time you had the sighting in it, you record the geographic positions, you can record, and then you also have your survey tracks, where you went. And that's very important because you want to know not only where you saw animals, but where did you spend effort and you didn't see animals. And that's very important to know. So keeping your survey tracks is highly important, and we incorporate all that in. And then there's all the typical GIS sort of functions attributed to here, where we have, when we have multiple records of environment, when we have multiple records of geographic positions, but you're pulling into a GIS system, maybe you only include the first one, last one, or all of them. And also you want to link your environmental data. So all that can be linked into a GIS file and then go to other applications. And then we include sort of a standard analysis package where you summarize your data, like how many individuals are in your database, how many are new as you're going through time, and then how many are marginal, how many resightings, min resightings, mean, max resightings. And then we have the concept of aspects. And so for like gray whales, we have their left sides or right sides are identifiable, also their flukes, and sometimes we use the dorsal fluke. So we can use multiple aspects of that individual to re-identify them. And then we can generate things like a discovery curve, which is very important, or how many individuals seen per year, or look at resighting histories. But we realized that this isn't enough, so we recently included what we call Discover R. And this is an interaction between Discovery and the R statistical package. And this allows to use all the functionality of R for Discovery. So what it essentially does is it takes your database and it provides it as a data frame to R, and then you can do various functions. And this is totally dynamic, and users can create their own functions or do whatever they want. So we're hoping that a lot of people will contribute here and contribute R functions in the future and do analysis to help users. And we already included some standard functions like the discovery curve, where this one's illustrating the number of individuals per year. And then you present that to the user as a graphical or a table output. And there's also various quick plotting. So I can select two different variables here if I want to look at latitude and longitude and how they interact, and do a scatter plot, or look at behavior as a function of group size, or you can do a box plot really quick, and you can look at your data very quick. And I think that quick assessment of your data constantly really helps researchers to understand their animals much better, and also the analytical tools that are needed. So this is how we're sort of developing this as we have our base scripts that interact with R and tell R what the setup is. This is all open source, so you can look at it and everybody can understand exactly what Discovery is doing with your data, and you can develop your custom scripts here. And once you develop it, you can just put a little thing there, and it shows up in Discovery, and you can run that function that you created anytime you want. And this is just an example of a script where you have a simple function here to do resightings. And anybody can just look at that and see what it's doing. And so here's an example of using our component with the discovery curve. Here's an example where we're doing reciting frequencies, which looks a bit psychedelic, but but the color coding here is actually by individual. So if I saw one individual 20 times, it would be all one color. But you can see our sighting has many different individuals through different times. So it's just, but you can change that. It's totally open source. You can get rid of it if you like, or whatever. And then you can use, the power of ours. It has so many packages. So you can just basically take it and plot it in Google Maps. And there's a function that provides that. So that's all our sighting data for the humpback dolphins in Hong Kong. And that's just a click of a button. And then you can look at activity budgets that you already have from your sighting data. And here we have most of the individuals in the area are foraging, and you can combine that relatively rapidly. Okay.
So other part of Discovery is actually to provide an interface between your data base and all the other analytical tools that you would like to use, but or you want to use output files that function as input files to other softwares. And in this case, for instance, Mark, where you can generate. Everybody who worked on Mark knows how painful it is to prepare this input files of all the sighting histories of zeros and ones, and it takes a while, and it's one of these tedious, silly things one has to do. Well, it can be generated very quickly with Discovery, and it can also be customized to how you want to do it. So you can decide which will be your capture occasion. Every encounter, or you pull your encounters and you decide what your capture occasions are. You include your covariates. You can also generate things for multi-state models, where you would replace the 0-1 matrix with states. In this case, this A and B, which could be reproductive stage, could be study site, could be breeding versus foraging sites, and
breeding versus non-breeding.
Versus non-breeding sites, and so on. Right. Foraging sites and so on. So actually make it this marker, like a basic marker capture, mark analysis. You can go further, and you can generate irregular capture occasions for open population models. You can also do your input file for robust design, when you actually decide which one is your primary capture occasion, which ones are your secondary capture occasions, and then you go. And again, everybody who was working with robust design knows how painful this process is before you actually start playing with actual getting some results. I think we are pretty good on time, so I don't need to speak that fast now. Another program that many of us working on individual identification like to use is SOCPROC, designed by Hal Whitehead Lab in Dalhousie in Canada. So here again we can generate output files, which are the input files to SOCPROC. We can have a couple of modes with very simple SOCPROC, and then more custom modes. We can actually include also locations where you can increase the number of analysis in terms of movement analysis, for instance, between research sites and so on, or the movement through continuous space. So we generate the primary files and then the secondary files with covariates when you include things like the in primary files we include things like image quality, distinctiveness of individuals, which then you can select later to put the restrictions on discovery on SOCPROC analysis. And the same for supplementary files with sex, age, and so on. And we generate also the export files for things like JS softwares, MarkMap or Quantum GIS or pretty much the GIS softwares out there, with citing these informations in the individual records with all the associated data that has already been included in discovery or entering the citing info at the first place. And now our number of users is considerable. It's increasing. It's not yet humongously huge, but it's increasing. And what is nice, which we just learned, that we have our keynote seal among them. That was nice. We didn't know it until yesterday. Obviously, you didn't use our forum. So.
Where we put it? Yeah, that we have no idea that we have that kind of forum.
It's good. It's there. And what is nice is actually number of terrestrial species that has been. It was designed principally for use at the first place for dolphins and whales, but then we adjusted it to make it really multi-species for both marine and terrestrial critters. And what is nice to see that more terrestrial species are being included. Just recently I heard of placebox and bontobox in Africa being used on discovery as well. We are going to use this example of Indo-Pacific humpback dolphins and gray whales as a bit of a quick overview of case study application, just in a couple of next slides. So with Indo-Pacific humpback whales, our study site based in Hong Kong represents only a fraction, tiny fraction, of the Pearl River Delta system, and what this chart represents is actually less than a third of our study site. It extends further to the west for another about 150 kilometers, so it's pretty substantial, and it's not only across a larger area spatially, it's also across a political border. Its administrative border is actually much more political than administrative, and it comes with implications. So for a number of reasons, we need to use multiple research teams. So the case of one study site, one species, multiple researchers. So obviously a coordinated collaboration and data sharing is critical. So in our case, we import everything to one centralized database, and in this database, we'll end up having some individuals that we do cross-matching. And in this, all the data is imported to one centralized database, and then cross-matches between individual databases from individual teams are being run. So we may have an individual 001 in Hong Kong, which is actually the same individual as 0100 in other study site in Zhuhai, just across the administrative border. And we can have this in this centralized catalog, while at the same time keeping them in different folders in individual databases for the specific study site. So that comes very, very handy. Well, quite important, not just handy. And gray whales, well, that's landscapes.
So as I said, we designed this initially to solve our own problems, but we think it's useful for other people as well. And Leszek demonstrated his problems, And I'll give an example of ours. I study a lot of the gray whale individuals with the western gray whale, which is a critically endangered population. And there's two populations of gray whales. We have the eastern population that breed down in Baja, but then migrate all the way up to the Boeing Chukchi Sea area. And so this population is actually the first species to be delisted off the Marine Mammal Species Act, and they're doing quite well. But the western population is critically endangered at about 150 individuals with 33 reproductive females, so very small population. And all we know about them is that they feed here, and they're genetically isolated, and we had the great hypothesis that they're genetically isolated because of this Kamchatka barrier. It offers a geographic barrier between the two populations. But then Bruce May came out and fooled us all, and told us that we were all incorrect, because that individual he tagged with the satellite tag off of Sakhalin, the feeding area, actually went all the way over to the eastern population. So now we're still scratching our heads what this actually means, because we have genetic data saying they're distinct populations, but they're spatially overlapping. But what this really does is cause a huge issue, and really begs for multiple researchers to collaborate. And this is where discovery comes very handy, because we have to work with different research groups all along this area to really pinpoint, what those individuals, where they occur through time, and that's very important aspect. And we have, photographic stranding data in Japan from incidental catches, and so that's very important to understand the gray whale species and also their social structure as a whole. And that requires not just one research group, but multiple research groups sharing huge amounts of data. And I think that's very important. So that's an example of our catalog as we presented before, where we just have these different links and we can cross-compare. And I don't know if you have how much experience with different research groups. It takes a huge amount of time looking at different data. So as much as we can try to standardize, it would be very beneficial. As I mentioned, we didn't try to reinvent the wheel here, and what we really are focused on is the data management aspects. And we realize that there's a lot of tools, and I'm really excited about this workshop, because it's bringing those tools together. And we didn't have that collaboration. We've been kind of in our own little boxes everywhere else, and I really am excited to bring all these other tools. And I can see, interactions between these different systems, like the IBIS, the I3S, Hotspotter. We could, potentially say, okay, you want to match with this system or that system, you're doing with this species or that species. Because as we mentioned, those algorithms are species specific in many cases. Sometimes they work on one species, sometimes they don't work on another. But we really need a broad application for seals that we didn't initially think that was going to be useful for, but they use it and it's good. And for African elephants, it's really exciting that people use it. And so we can see that we can put that in and collaborate to a great degree. So I love to see this convergence of science come together. And then there's the other applications like, as Chuck mentioned yesterday, the WildMe, which is more of an online platform. So if you want to just take your data and you want to share it online with other people, you can have an export function and just say, put it online, and here's my sighting data, and interact with more people. So I love to see this convergence of science and this workshop that put it together, that we can all discuss and find ways, and I hope to discuss that later on in our session today. So this is our online forum. Discovery is totally free, available to anyone. We have our website, and then we have our forum, which people didn't realize we had, but but this was our great attempt to I think John mentioned it yesterday, to avoid a bunch of emails, but we weren't that successful because people don't know there's a forum here, and so. But it's to share the knowledge, share experiences. If you have issues, there's bugs, which I didn't know you guys had. Tell us, and we'll try our best. This is, just this. We do this for our own reasons, and we have limited funding, but we try to support it as much as possible, and hope hopefully the software evolve over time.
Before you go to the next one. Just to mention, there is we just made an update with a new version of Discovery, literally a couple of weeks ago. So what you have been downloading now is kind of outdated. There is a new version that will be placed here with new manual. The manual is pretty much ready. The new version with R incorporated will probably be out online within next couple of weeks, less than a month. So you might want to revisit the site if you're interested to go further.
So Leszek and I are the primary principal investigators here, but we try to use and abuse students as much as possible, and that they have collaborated quite much and have helped out quite a bit, and they use it and have provided valuable feedback and support for like the manuals and all that type of information and potential errors they've had. And we have also had some people interact in the forum and email us, so we appreciate that feedback, and we constantly are trying to integrate our system.
Stephen and Carmen are here in the audience, and they really did a lot of hard work on testing all the stuff that you guys end up with a bit less bugs than there would be otherwise. So that was a tremendous help. Thank you.
And with that, we'd like to thank our home institutions and our funding organizations that have supported this. And I think, as I mentioned, we're very limited in funding, but we try our best as much as possible. Thank you.
Yep.